Hi everyone, in this video we're going to take a look at some examples of epithelial tissue. Now before we jump into the identification part, I want to do a brief overview of the characteristics and how we classify epithelial tissue. So there are four main characteristics you need to remember when we're talking about epithelial tissue. First off, the cells are always closely packed together. There are many junctions holding the adjacent cells together. They form rows and columns. There's very little extracellular matrix between each cell. Secondly, we can identify the sides of the cell by using these terms apical surface, lateral surface, and basal surface. Now the apical surface is always the free edge. It's going to be a side of epithelial tissue that's not attached to anything. There will be an open space there. The lateral surface is simply the sides of the cell. This is where you'll see the majority of the junctions holding these cells together. And the basal surface is simply the bottom layer of the ET. Underneath all epithelial tissue is connective tissue, and the basal surface is going to be the bottom layer of the epithelial tissue that's attached to a region called the basement membrane, and that basement membrane serves as your attachment point to the connective tissue below that ET. Thirdly, all epithelial tissue is avascular, which just simply means there's no blood supply. There's no blood vessels, no veins, no arteries, no capillaries are going to be found in epithelial tissue. Those cells get their nourishment by the surrounding connective tissue. Most connective tissue is very vascular. So by diffusion and osmosis and all of these other membrane transport mechanisms that you've learned about, the epithelial tissue cells can get the nourishment that they need. They do not have a direct blood supply. And then lastly, epithelial tissue is actively mitotic, which just means that these cells undergo mitosis constantly. A lot of epithelial tissue is found in areas where there's friction, so we lose these cells easily. And being actively mitotic allows these cells to be replaced quickly. Now, when naming epithelial tissue, most will have at least two names. The first name tells us how many layers there are, and we can have a simple epithelial tissue, which means one layer, or a stratified epithelial tissue, which means two layers. And the second name tells us the shape of the cells. Squamous means flat or squished together. And if I was to draw a squamous cell, it would kind of look like a flat pancake, something similar to this, where these cells are touching all in a row. And since the cell is very flattened and scale-like, the nucleus will also be very flattened and scale-like. So you'll kind of see a squished nucleus in there as well. Cuboidal is going to look like a cube, so these cells are going to be box-shaped. And since the cells are a bit bigger and they're not squished, you'll typically see a nice round nucleus inside of these cuboidal cells. And lastly are the columnars. Columnar cells are going to form columns. These are going to be very tall cells like so, and that nucleus will almost look kind of like an oval because it stretches out within that column as well. So these little lines that I'm putting inside the cell represents the nucleus in each one. So if we put these classification categories together, we can have a simple squamous epithelial tissue, or we could have a simple cuboidal or a simple columnar. And likewise, we could have many layers. So we could have stratified squamous or stratified cuboidal and stratified columnar. Now on the following slides, we're going to see a series of photos taken using our microscopes in the lab. And one thing you need to be able to do is to recognize each epithelial tissue example by looking at that picture. Now to start with, we're going to go through the simple epithelial tissues. So our first example is of simple squamous. So this is going to be one layer of very flattened cells. So it's going to look something similar to this, where I'll have a cell and then a nucleus that's kind of squished in it, another cell and a nucleus that's squished, and another cell and a nucleus that's squished. Okay? Now, believe it or not, both of these images that you're looking at is of the same tissue type. We're just looking at it from two different viewpoints. So let's look at the photo on the left first. Where you see that pointer coming down from the microscope lens, we're actually pointing at the simple squamous epithelial tissue. And it is this very thin layer that's right around this area, and it runs in a circle. I'm going to kind of come around and circle it to bring your eye to it. So there to here. I'm going to try my best to circle it around. Right in there. Now, in between, kind of where I've tried to draw that circle, attempted to draw that circle, that's going to be your epithelial tissue. So let me draw an arrow pointing at this squished nucleus, and maybe you'll see what I'm talking about. So I'm going to put an arrow right here. You see that squished nucleus right there? And then I'll put another arrow right here, and then another arrow right here, and then another arrow right down here at the bottom. And as we work our way around, if we look closely, we can start to see some squished nuclei in here. So what's happening is this layer of simple squamous ET is running in a circle around this group of cells that we see in the center. And this free area, this free space that's between that simple squamous and then that group in the middle is the apical surface. Now I'm going to clean up my drawing a little bit here. There we go. And get rid of these lines here so that we can actually see maybe a little better that simple squamous. I wanted to draw those lines, that little circle, just to bring your eye to the layer of simple squamous ET. So now that I've got it cleaned up a little bit, let me draw an arrow specifically to that layer, kind of a big one, right here. Okay, so you can see if we follow my little dotted line around, this is our layer of simple squamous epithelial tissue. You can see those flattened nuclei, and they run all the way around. Now the picture to the right is the same tissue type. There's a different type of stain, so you see it's a different color, but it's the exact same tissue type. And what we're looking at to the right is called a surface view. So imagine, I'm going to borrow my daughter's notebook right here. So imagine that we have got the sectional view, which is what we see over here on the left, simple squamous section. We're looking at that layer kind of like this, where you can see how thin it is and looking at the edge. The picture to the right is called a surface view. And that surface view is more like this, where you're looking at the surface of the cells rather than a sectional view so that you can see how thin they are. The picture to the right shows you how tightly packed these cells are and how there's little to no extracellular matrix that's between them. There's many junctions that are holding them together. Now, simple squamous ET is the thinnest tissue in the body. You have this wherever we need exchanges to happen easily and quickly. So in the kidney tubules, where your kidneys are not tubules, but the filtration unit in your kidneys, where the blood gets filtered, that simple squamous ET, you have this tissue type in the air sacs in the lungs and also the capillaries, so that gas exchange can take place very easily. Next up is simple cuboidal epithelial tissue. Now, simple cuboidals, one layer of cube-shaped cells, and a key characteristic to these is that a lot of times your cuboidals will form a circular structure. So it'll look something similar to this, where you may have a cube, and then another cube, and another cube, and you kind of see how it's starting to form this circular structure. And your apical surface, or your free surface, winds up in the middle. I'm going to put an A right here in the middle. So your center is the apical surface, or the free surface that's not touching anything. And then your cuboidal cells form a circle around that apical surface. And since our cell is much bigger than what a squamous is, you'll also have a nice round plump nucleus inside of that. Now, if we look at this photo, you see this exact same pattern. There are many circles. These cuboidal cells are forming circles. And the big purple circle that you see within the pink cells, that's the nucleus that we see. So I'm going to circle a couple of these just to bring your eye to it. Notice this area right here. Okay? This purple circle, that's a nucleus. And my apical surface is here in the middle. I'm going to put an A there. That's my apical surface. Here's another circular group of cuboidals. Here's another circular group of cuboidals, and here, and here, and here, and honestly, they're all over the microscope slide. They are everywhere. All of these groups of circles that you see, I usually compare them to a donut. Since the apical surface is in the center, it's kind of like the whole of your donut, and then the cells form this circular formation around it. So all of this is simple cuboidal epithelial tissue with that apical surface in the center. Now, you find simple cuboidal in a variety of places. A lot of glands are made of this tissue type. There's a layer around the ovaries in females. You can also find this in the kidney tubules, little tiny tubes that are in your kidneys that will receive the filtrate after the simple squamous filtering unit does its job. That filtrate will pass through the kidney tubules, which is made of this simple cuboidal ET. Now we've come to simple columnar epithelial tissue. Now, simple columnar is one layer of very column-like cells, so tall cells. We'll typically see this formation where we've got a column and then another and then another and so on, and we'll see that nucleus inside of each columnar cell. Now, simple columnar has some variations to it. One of the variations can be the presence of cilia. So it's possible to have ciliated simple columnar, in which case you will have these column-like cells all in a row, and then there'll be these little hair-like structures on the surface. Now remember, we learned this from chapter 3, cilia are these hair-like projections that move in a sweeping motion, and they move structures past their surface. Inside the female reproductive tract, the fallopian tubes, or uterine tubes, sometimes they're called, are actually lined with ciliated simple columnar epithelial tissue. Now, not all simple columnar has the cilia. There's another type called non-ciliated simple columnar, and that's actually what you're looking at here in this photo, is non-ciliated simple columnar. And a characteristic of non-ciliated is a lot of times it will have microvilli on it. Microvilli are these finger-like projections that increase surface area for absorption. So microvilli with the simple columnar would look kind of like this. We've got our column, and then there would be these fingers, and then our next column with these fingers, and then our next column with these fingers, and so on. They're different from cilia, okay? Microvilli are very different. These are extensions of the plasma membrane, and they're going to be used to make more surface area for absorption. Microvilli are not easy to see under the microscope, so a lot of times you won't notice the microvilli if it's present on the columnar cells. Cilia, on the other hand, is easy to see. You can see these little wispies, these little hairs on the surface of the cells. Now, in our microscope slide that we're looking at here, this is non-ciliated simple columnar. So there's no cilia present on these, and it actually does have the microvilli, but again, it's not that easy to see. Another characteristic, aside from having cilia or microvilli, is the presence of goblet cells. A goblet cell is a single-celled gland that produces mucus, and goblet cells look like little bubbles. Now I'm going to draw your eye, I'm going to draw on this microscope, so hopefully to bring your attention to this row of columnar cells. So I'm going to choose this spot here because it's pretty easy to see. And what we're looking at here in this picture is just many of these columns. I'm going to try to highlight a few of them, or maybe outline a few of them. So there's a column, there's another one, there's one. Can you kind of see, hopefully maybe, you can see these columns all kind of working their way deep down into close to the connective tissue, which is here at the bottom. So all of these little rectangles, I'm trying to outline those columnar cells so that you'll know exactly what you're looking at. And the goblets that I mentioned, they're going to look like little bubbles. So all of these little clear spaces that you see, there's one, there's one, there's one. There's a big cluster of them right around in here. And then some over here in this area, and some over here in this area. They're everywhere. These goblets are going to be producing mucus. Now this particular slide's taken from the small intestine, so this is going to be non-ciliated simple columnar epithelial tissue, and it does have microvilli on it, even though it may not quite be obvious to see. Microvilli is present here, and all of this that you see kind of going around in an arch is your columnar ET, and down here in the middle where you can see the pattern changes right here, this is actually connective tissue. Now we've come to pseudostratified columnar epithelial tissue. Pseudostratified is a modification of our simple columnar. It's going to appear stratified because the nuclei can be at different levels in the cells, but it's really a simple epithelial tissue. So that's why we have that prefix pseudo here. That prefix pseudo means false. So falsely stratified, or it gives the appearance of being stratified when it's really not. Now, here in the photo, you're looking at pseudostratified columnar, and a key characteristic that is almost obvious from the get-go is that there's cilia present on that apical surface. So this whole top area that's not touching anything, this is what we would call our apical surface. And then you'll notice there are these little wispies, these little hairs coming off the top. These little wispies are cilia. And all pseudostratified columnar has cilia on it. Okay, so that's a kind of a giveaway. If you're seeing ciliated cells, it's going to be a columnar ET, and probably it will be a pseudostratified. And we also can tell it's columnar, not only because it's quite tall, but there's goblet cells that are present. Goblets, again, are these little bubble-like cells that secrete mucus. I'm going to circle a few of them here. So there's a goblet, mini goblets right around here in this area. They take up most of the space, actually. All of those that I have circled are your goblet cells. So this is our pseudostratified columnar, and down here at the bottom where you can see the pattern changes, this is connective tissue. So that's going to be CT, and then this area here where that pointer is, this will be our epithelial tissue layer that is that pseudostratified columnar. The main place where you find this in the body is in the respiratory system. Your nasal cavity, your trachea, even down into the bronchi, they're all lined with this pseudostratified columnar. And that cilia is moving the mucus kind of up and out of our lungs, so that we can get rid of anything that's contaminated that we've inhaled and we didn't even know it. It gets trapped in that mucus, and then we're able to get rid of that. Now we're moving into the stratified epithelial tissues. Now the stratifieds are going to have many layers to them, and first up is stratified squamous. Stratified squamous is found in areas where there's a lot of friction, so there are many layers found in this type of epithelial tissue. Now there are two variations to the stratified squamous epithelials. The one on the left you're seeing is called non-keratinized stratified squamous, and the photo to the right is showing a different kind called keratinized stratified squamous. Now what sets these two apart is the presence of keratin on the surface of the keratinized stratified squamous. The non-keratinized does not have that outer layer of keratin. So looking at the left photo, our apical surface is here where nothing is touching, so all of this side to that epithelial tissue is going to be our apical surface of the cells. And we're seeing many layers, actually all the way in our field of view. Everything we're seeing is going to be stratified squamous epithelial cells. The cells are very flattened, and if you look on the surface where that apical these arrows are, that when I was showing you the apical surface, you can see some flattened cells like right here, and then that squished nucleus that's right there. And then here is another one that's pretty flattened, and another one here that's pretty flattened. Now as we go down, you can see some that may be a little bit larger, but if you continue to look, there are many flattened or squished squamous cells intermingled throughout this entire tissue. The photo to the right is also a stratified squamous, but there is an outer layer of keratin on the surface. From here all the way down to here, All of this is going to be our stratified squamous epithelial tissue. And down here at the bottom, where you see this light pink and the pattern changes a little bit, this is connective tissue down here at the bottom. Now, the stratified squamous cells are here. This is our stratified squamous layer, that kind of dark pink where you see these hills and valleys dipping around. That's our stratified squamous layer. This purple layer here, that is our keratinized layer. There's a very thick layer of keratin on the surface. These keratinized cells are actually dead cells that before they died produced a lot of keratin. And as they worked their way up to the surface and were replaced by cells undergoing mitosis along the basement membrane down here along the bottom, they got so far away from the blood supply and the connective tissue that they died. So these cells here, where I have my bottom arrow, these are living squamous cells. These cells are actually called keratinocytes. That's the name of your skin cells. This is a skin slide that we're looking at here to the right. These cells are called keratinocytes, and they produce keratin. And as they work their way up, remember mitosis is happening down here on that basement membrane along that bottom layer. So as mitosis is happening and these cells are getting pushed further and further up, they're getting so far away from that blood supply and the connective tissue that they die. So these outermost cells, these are actually dead cells, and we describe them as being keratinized because essentially they're just cells full of keratin. They're dead. They're not functional anymore. They're not really doing anything. They're just sitting there full of keratin. And it puts this outer layer of keratin on the surface of our skin, which is a very strong structural protein. So it's a good protective layer. It's also a waterproofing layer. It prevents water loss from the surface and prevents too much water from coming in from the surface as well. So we don't lose water from our lower layers, and we don't take on too much water from our outer layers because of that presence of keratin there on the surface. So both of these are stratified squamous. They're very deep. We have these in areas where there's a lot of friction. So I already mentioned the skin over here with keratinized stratified squamous. The skin is where we find this mostly. Our hair, our nails is all made of keratinized stratified squamous. The non-keratinized is found within our mouth and in our esophagus, because these are areas that's under a lot of friction as well. And the vagina is lined with non-keratinized stratified squamous as well in the female reproductive tract. Next up is stratified cuboidal epithelial tissue. Stratified cuboidal is the rarest epithelial tissue in the body. It's not found in a whole lot of places. You mainly have stratified cuboidal in large glands. There are some glands in the male reproductive tract that are composed of stratified cuboidal, and sweat glands within our skins also composed of stratified cuboidal. Now, this is going to have the same layout as the simple cuboidal, where we have these cube-shaped cells forming a circle, and our apical surface is going to be in the middle. So right here, you can kind of see this donut-like structure with our apical surface in the center. I'm going to put an A there for that apical surface. And you can tell there's more than one layer of cuboidal cells here. We've got a pretty obvious layer right here, then there's another layer here, and as we get down here in the bottom, there's a third layer here. So typically you'll see 2 to 3 layers of cells in stratified cuboidal epithelial tissue. Our last epithelial tissue is called transitional. Transitional is a type of stratified epithelial tissue, and it gets its name because it's found in organs that stretch. So it literally transitions, it changes depending upon the size of the organ. The main location you need to recognize for transitional is the urinary bladder. Now, as the urinary bladder fills up with urine, it stretches out, and these cells can start to flatten out and look a lot like stratified squamous. Then when we empty the bladder, the cells get larger because they're not squished anymore. So they'll kind of recoil back to their original position. And in that case, they'll look like much larger plump cells. So here in the photo, our apical surface is all of this free edge that we see, whoops, apical, if I can spell here, let me rewrite that. There we go, apical. Now this is all of this area, the surface, and then the cells that you see from that apical surface down is going to be our transitional. And if you look under, there's some kind of pink waves. You see it right in here. This is an area where the pattern changes a little bit. This is going to be connective tissue. So you see some CT here, CT at the bottom, and then right around here in the edge, that's going to be connective tissue. So that means everything from here to where that pattern changes is going to be our epithelial tissue. Now, one key characteristic to transitional is to recognize the pattern. Transitional tends to form these hills and valleys. So you can see it kind of running down from the left to the right of your of the photo here, and then it curls and kind of comes back up to the left. That's a key characteristic to transitional. It's always going to kind of have this rounded or hills and valleys type structure.